Dear students, in this video I'll be discussing the concept of endomembrane system that comes in chapter 8 cell in class 11. If you take endomembrane system, it's a system in which the functions of different cell organelles are coordinated. The type of cell organelles that have been coordinated, one will be the endoplasmic reticulum, the another one is Golgi complex, the another one is lysosome as well as vacuole. All these structures, endoplasmic reticulum, Golgi apparatus, lysosomes and also vacuole, they are all single membrane bound cell organelles. Their functions have been coordinated. Any cell organelles which are in which the function has been coordinated, then together what we call it as endomembrane system. Here the endoplasmic reticulum involved in formation of proteins, that is particularly the rough endoplasmic reticulum. This is the ribosome, endoplasmic reticulum associated with ribosome that we call it as rough endoplasmic reticulum. It is involved in production of protein. So whatever the protein that is produced, it will be produced. It should be sent outside. Then it enters into the vesicle. It enters into the vesicle. Then properly it should be packed. For that purpose, it goes to the Golgi apparatus. Inside the Golgi apparatus, the packaging takes place. Then later, whatever the vesicle that has been released from this Golgi apparatus, that fuses with the cell membrane, and the content will be released outside. The content will be released outside. In the same way, even the lysosomes have been produced by the Golgi apparatus. So the functioning of the endoplasmic reticulum as well as the Golgi apparatus, even lysosome, including vacuole, some components or any protein that should be stored, even that will be sent to the vacuole, particularly in plant cells. All these cell organelles, endoplasmic reticulum, Golgi apparatus, lysosome, vacuole, their function has been coordinated. Such a system, what we call it as endomembrane system. So we'll be studying about endoplasmic reticulum. Once we complete endoplasmic reticulum, I'm going to take few MCQs. Then later we'll discuss about Golgi apparatus. Even in this, we'll discuss some MCQs. Then even lysosome as well as vacuole. So if you take the endomembrane system, the cell organelles that are involved. Just now I told that it is endoplasmic reticulum, Golgi apparatus, then lysosomes as well as vacuole. The cell organelles that are not involved in endomembrane system, it is mitochondria, chloroplast as well as peroxisomes. First we'll take about the single membrane bound cell organelle as well as double membrane bound cell organelles. So if you take the vacuole, lysosome, Golgi apparatus, endoplasmic reticulum, all these are covered by a single membrane. That's why we call them as single membrane bound organelles. If you take nucleus, mitochondria as well as chloroplast, they have been covered by two membranes. That's why we call them as double membrane bound cell organelles. So in this we'll take the first cell organelle that is involved in endomembrane system. It is endoplasmic reticulum. So there is the structure of endoplasmic reticulum. You can see that the nucleus, there is the nucleus. Then the nucleus is connected to the rough endoplasmic reticulum. It is continuous with this rough endoplasmic reticulum. On the nucleus you find the openings. These openings are called nuclear pores. But in this video we'll be discussing about only these cell organelles. I will not take the structure of nucleus. If you take endoplasmic reticulum, it is a network made up of small tubules that is scattered all over the cytoplasm. It can act as a supporting structure. It acts as a cytoskeleton. It's a single membrane bound cell organelle. But this cell organelle, it is the largest compartment. Compartmentalization is one of the important characteristics of the eukaryotes. The compartments are mainly because of presence of different types of membrane bound cell organelles. It can be mitochondria, it can be chloroplast, even endoplasmic reticulum, Golgi apparatus. All these are different cell organelles. All these cell organelles have certain compartments. Such type of compartments are present only in eukaryotes. Such type of compartments are absent in prokaryotes because membrane bound cell organelles are absent in prokaryotes. So if you take the largest the compartment formed by a particular cell organelle, it has nothing but it has nothing but endoplasmic reticulum. First, let us understand about the two types of compartment that has been created by this endoplasmic reticulum. So it has divided or it has two distinct compartments. You can see on the screen. That is, one is luminal compartment, whatever the space that is present inside the endoplasmic reticulum, that we call it as luminal compartment. Then outside the endoplasmic reticulum, that is cytoplasm, it represents the extraluminal compartment. So endoplasmic reticulum divides, or it has two compartments. One is within the endoplasmic reticulum, the another one is outside the endoplasmic reticulum. The compartment present inside the endoplasmic reticulum, it is luminal compartment. Then outside the cytoplasm, that represents the extraluminal compartment. And also we'll take the types of the endoplasmic reticulum. So one will be the rough endoplasmic reticulum, the another one is smooth endoplasmic reticulum. Rough endoplasmic reticulum, that is associated with the ribosome. So these spherical structures, they represent the ribosome. Represent ribosome. If the endoplasmic reticulum associated with ribosome, then we call it as rough endoplasmic reticulum. You can see here, the endoplasmic reticulum is not associated with ribosome, then we call it as smooth endoplasmic reticulum. So if you take the functioning of this endoplasmic reticulum, the functioning, the rough endoplasmic reticulum is actually involved in synthesis of protein, and also it is involved in secretion. Actually, the ribosomes are involved in synthesis of protein. Such type of proteins, if they are required outside, then those proteins will enter into the endoplasmic reticulum. Then later they will be sent to the Golgi apparatus. From Golgi apparatus, it will be sent outside. So rough endoplasmic reticulum involved in protein synthesis and also involved in secretion. Smooth endoplasmic reticulum, it has many functions. Smooth endoplasmic reticulum actually involved in synthesis of lipid. That is also involved in formation of membrane lipids. Phospholipids are produced. Even the smooth endoplasmic reticulum is involved in production of steroidal hormones, that is in animals. Along with that, in animals, it is involved in detoxification. It is going to neutralize the toxic substances that are present in our body. That is the major function of smooth endoplasmic reticulum. So if you take the functioning, rough endoplasmic reticulum, protein synthesis. Smooth endoplasmic reticulum, it is involved in synthesis of lipid, and also it is involved in synthesis of steroidal hormones. Along with that, in animals, it is involved in detoxification. The detoxification, where the neutralization of the toxic substances takes place, it happens in liver. So liver cells has more number of smooth endoplasmic reticulum. So these are all the information what is required for your board exams. But for competitive exams, you need extra information, some related information. So all those information I have taken in this multiple choice questions. So the first question, which of the following is the largest single membrane bound intracellular compartment? Just now we have studied that endoplasmic reticulum has two compartments. One is luminal compartment, the another one is extraluminal compartment. Extraluminal compartment, it is a cytoplasm. Then luminal compartment within the endoplasmic reticulum. Even the Golgi apparatus, it has two compartments, outside the Golgi apparatus as well as inside the Golgi apparatus. But here they are asking about the largest single membrane bound intracellular compartment. It should be the endoplasmic reticulum. The answer should be the fourth one. This is the answer. Then the ribosome, it is not at all a single membrane bound cell organelle. It is a non-membrane bound cell organelle. It is non-membrane. It's a non-membrane bound cell organelle, but the nucleus, it is double membrane. Double membrane bound cell organelle. Golgi apparatus, even it is single membrane bound cell organelle. But here they are asking about the largest intracellular compartment. It is endoplasmic reticulum. Coming to the second question, which of the following biomolecule are not synthesized by the endoplasmic reticulum? Just now we have studied that endoplasmic reticulum involved in synthesis of protein, involved in synthesis of lipids, and also involved in synthesis of steroidal hormones, that is cholesterol. So the answer will be, it is the nucleic acid. Nucleic acids are not synthesized in endoplasmic reticulum. Then if we take the third question, microsome, the smaller endoplasmic reticulum or small vesicle that are produced from the endoplasmic reticulum or fragmented endoplasmic reticulum, that we call them as microsomes. They can be rough microsome as well as smooth microsome. So microsome represents, it will not represent, or it is not the compartment of Golgi apparatus. It is not the ribosome, and also it is small endoplasmic reticulum compartment. It is wrong. It is a small vesicle fragmented endoplasmic reticulum. So the answer should be D. Name the antibiotic which inhibit the synthesis of oligosaccharides. Oligosaccharides are also synthesized inside the endoplasmic reticulum. There will be addition of the carbohydrate to the protein that are produced in rough endoplasmic reticulum, but that will be inhibited by one particular type of antibiotic. So that antibiotic, it is actually produced by a bacterium. It is the antibiotic is tunicamycin. It is tunicamycin. That is the antibiotic that inhibit synthesis of oligosaccharides inside the endoplasmic reticulum. Name the site where the detoxification of xenobiotic. Xenobiotic means the outside our body. Anything that comes into our body, that such type of chemicals we call them as xenobiotic. Synthetic chemical that is foreign to the body, they are called xenobiotic compounds. The xenobiotic compounds, it can be the alcohol also. Such type of alcohol, or it might be certain narcotic drugs. All they are toxic to our body. They should be neutralized. That neutralization of toxic substances, we call it as detoxification. The detoxification actually takes place in smooth endoplasmic reticulum. So the answer is a C. Then coming to the next one, which of the following coated vesicle transport protein from endoplasmic reticulum to Golgi apparatus? So if you take that the nucleus, for example, there is the nucleus, then surrounding this nucleus, you find the endoplasmic reticulum, then you find Golgi apparatus. So the convex region of Golgi apparatus is present towards the endoplasmic reticulum. The concave region of Golgi apparatus is present toward the cell membrane. For example, there is a cell membrane. Here you find the ribosomes. So ribosome, it is involved in synthesis of protein. So whatever the protein that has been synthesized, that enters into endoplasmic reticulum. Then inside the endoplasmic reticulum, with the help of cysteine residues, the modification takes place. The protein becomes stable by the formation of disulfide bond. That disulfide bond is mainly because of cysteine residues. Cysteine is nothing but an amino acid that provides a disulfide bond. Once it is formed, it will be sent outside. It will be sent outside. A vesicle will be formed, but that vesicle should be coated by a protein. That is the coat protein complex. The coated complex protein, that is, we call it as COP. So it forms an outer covering. So that later, that vesicle that is covered by COP2 protein, protein. So that that helps in transportation of this vesicle to the Golgi apparatus. So here you find the membrane of endoplasmic reticulum. Now it reaches the Golgi apparatus. So this covering, whatever they have given in the red color, that is nothing but COP. It is protein complex. That is complex protein 2. That helps in transportation of such type of vesicles to Golgi apparatus. So if you take the clethrine, it's a type of protein that helps in formation of endosomes. For example, if you take clethrine, the functioning of clethrine. So whenever a particle should enter into this cell, then there will be clethrine that helps in formation of endosome. So the plasma membrane invaginates. It invaginates and forms a vesicle. The formation of such type of vesicle mainly depends on such type of the clethrine. So that helps in formation of the vesicles, that is endosomes, whereas COP2, that core protein, it is required for the transportation of vesicles from endoplasmic reticulum to Golgi apparatus. So here the answer should be B. Clethrine, that is required for the formation of endosome. Then coming to the next one, detoxification of organic compound is carried out by dash Chronic use of compounds such as barbiturates. Barbiturates, they are the synthetic sleeping pills. The ethanol can lead to proliferation of dash type of cell organelle in the liver cells. Liver cells actually involved in detoxification. The type of endoplasmic reticulum involved in detoxification is smooth endoplasmic reticulum. When the person starts taking barbiturates or ethanol, the number of smooth endoplasmic reticulum keeps on increasing in liver cells. Then coming to the next question. Just now I told that the stability of the protein produced inside the endoplasmic reticulum, it depends on the formation of disulfide bond. So here we have given that a disulfide bond play an important role in the stability of proteins. When the protein enter into endoplasmic reticulum lumen, they contain dash in the reduced form, which is converted to oxidized disulfide bond when they leave the endoplasmic reticulum. For example, there is the endoplasmic reticulum. Then here you find the ribosome. This represents the ribosome. In the ribosome, you find smaller subunit as well as larger subunit. Both are involved in formation of protein. So the protein enters into the endoplasmic reticulum. For this, you find certain the channel proteins, translocon. They are called translocon. So I am going to show the translocon in brown color. So this represents the translocon. With help of translocon, the ribosome send the protein into the endoplasmic reticulum. Then inside the endoplasmic reticulum, the stabilization of protein takes place. For that purpose, the protein should fold. For the folding of the protein, disulfide bonds are required. For the formation of disulfide bond, a type of amino acid is required. The type of amino acid that is required, it is cysteine amino acid. So that cysteine amino acid will be present inside the endoplasmic reticulum. So stabilization of such type of protein, the blue color one whatever I have shown, that is actually the polypeptide chain that come from ribosome. Through translocon, it enters into the lumen of endoplasmic reticulum. Later, the stability of the protein depends on the formation of disulfide bond. That disulfide bond actually provided by cysteine residues. Then coming to the next one, the ninth one, sequester. Sequesters means which accumulate any component that we call it as sequester. Here sequesters calcium ions. The structure that accumulates calcium ions, particularly in skeletal muscle as well as in the cardiac muscle. So you find the sarcoplasmic reticulum and also you find the nucleus. There is a nucleus. It is multinucleated. Surrounding the nucleus you find the endoplasmic reticulum. This endoplasmic reticulum, it is smooth endoplasmic reticulum. Actually it stores calcium. So the contraction of muscle mainly depends on releasing of calcium. That calcium will be stored in this endoplasmic reticulum. Such endoplasmic reticulum that stores the calcium. That what we call, that is nothing but the smooth endoplasmic reticulum. The smooth endoplasmic reticulum present in muscle fiber, it is called sarcoplasmic reticulum. So the answer should be, for this, the answer should be sarcoplasmic reticulum. Then coming to the 10th question, in cells that secrete large amount of proteins, example the cells of kidney and salivary glands, more number of dash are present. The protein synthesis takes place in ribosome, particularly it is the ribosome that are found attached to the endoplasmic reticulum. So in kidney as well as in the salivary glands, the secretion will be more. That means they have more number of the ribosome attached to the endoplasmic reticulum. Such type of endoplasmic reticulum we call them as rough endoplasmic reticulum. So in kidney cells as well as salivary glands, you find rough endoplasmic reticulum. More number of rough endoplasmic reticulum. If you take liver cells actually involved in detoxification. That's why they have smooth endoplasmic reticulum. Coming to the 11th question, identify the wrong statement related to endoplasmic reticulum. Oxygenases. These are all the enzymes that are required for the detoxification, but they are present in smooth endoplasmic reticulum. But in the first statement, we have given rough endoplasmic reticulum. That's why the first statement is wrong. Wrong statement. If you take the second one, microsomes are the small vesicles formed by the breakdown of endoplasmic reticulum membrane into fragment. They are called microsomes. In that, if they are associated with ribosome, then they are called rough microsomes. Not associated with ribosome, then it is called smooth microsomes. The third one, the coat protein complex. Just now we have discussed it. The coat protein complex 2, or it is represented as COP, is a group of proteins that facilitate the formation of vesicles from the endoplasmic reticulum. So that goes to the Golgi apparatus. Such protein that are required for the formation of vesicle, that is required for the Golgi apparatus. That is what we call it as coat protein complex 2, that is abbreviated as COP2. So even this statement is correct. The membrane, if you take the fourth one, the membrane of rough endoplasmic reticulum and smooth endoplasmic reticulum continuous, it is correct. They are in a continuous pattern. Even the fourth one is correct. Translocon, just now I told that they are present in the membrane of endoplasmic reticulum, through which the protein that is produced in ribosome enters into the lumen of the endoplasmic reticulum. Such a protein which forms the channel that we call it as translocon. So translocon is a protein channel embedded in the membrane of endoplasmic reticulum, through which nascent polypeptide moves through from ribosome to the endoplasmic lumen. So even it's a right statement. So if you take a wrong statement here, only first statement is wrong. So the answer should be A. Let us take the Golgi apparatus. Golgi apparatus, even it is a single membrane bound cell organelle. The C-shaped structure, what we can see, we call them as cisternae, and these spherical structures are called vesicles. These cisternae are arranged one above the other. Then the Golgi apparatus has two phases. For example, there is the cell membrane. The concave region faces towards the cell membrane. Convex region faces toward the nucleus or endoplasmic reticulum. The convex region which faces the endoplasmic reticulum or the nucleus, that we call it a cis phase, formation phase. Then if you take the next one, the concave region that is present toward the plasma membrane, it is called the trans phase. It is also called maturation phase. Then what are the vesicles that are produced with the help of COP2 protein? For example, this will be the endoplasmic reticulum, and this is the vesicle that is produced. The vesicle that come from endoplasmic reticulum, that we call it as, that will be covered by, that is COP2 protein. Such type of vesicles, they reach the Golgi apparatus. They are called incoming transport vesicles. Now these incoming transport vesicles, they enter into lumen of Golgi apparatus, where the modification takes place, packaging takes place. From there, it will start entering from one cisternae to the another cisternae. Then later this will join to the cisternae. Then later it comes and it comes out of that Golgi apparatus, that particular phase, what we call it as trans phase. Then this the vesicle, now it joins with it fuses with the plasma membrane, then its contents are released. It will be released outside. Such vesicle that comes out of Golgi apparatus, that we call it as secretory vesicle, which joins the Golgi apparatus, that we call it as incoming transport vesicle. Then later that protein will be sent. That process what we call it as secretion. Such cell organelle was first identified and studied by Camillo Golgi. So scientist who first observed Golgi apparatus, it is the Camillo Golgi. That's why it is called Golgi apparatus. But here we have given double membrane. It is actually single membrane cell organelle. Then if you take the component, consists of flat disc shaped sacs called cisternae. Just now I told that these are the cisternae. They are arranged one above the other. Then cisternae are concentrically arranged near the endoplasmic reticulum. So it has two phases. One will be the convex cis phase, that is also called forming phase. Concave, it is also called trans phase. Trans phase, it is also called maturing phase. The maturing phase phases towards the plasma membrane. Cis phase phases toward the endoplasmic reticulum. So it has two functions. One will be the packaging of material. The next one is modification of the material. The packaging as well as modification will be the same. So here we have given material that should be delivered either to the intracellular targets or secreted outside the cell are packed in Golgi apparatus. Intracellular, within the cell itself. Material to be packaged in the vesicle come from the endoplasmic reticulum. So this represents the endoplasmic reticulum, and these spherical structures, they are nothing but ribosomes. So ribosome produces, so you can see that ribosome producing protein that enters into the lumen of endoplasmic reticulum. Later there will be formation of transport vesicle. The transport vesicle fuses with the Golgi apparatus at the cis phase. Then it moves from one cisternate to another cistern, and later it comes out as secretory vesicle. That secretory vesicle fuses with the plasma membrane, then it will be sent outside. So here both endoplasmic reticulum as well as Golgi apparatus involved in the secretion. So that is the functioning of Golgi apparatus. Due to this reason, Golgi apparatus is always near the endoplasmic reticulum. Then whatever the protein that has been produced in endoplasmic reticulum, they are linked with the carbohydrate. That what we call it as modification. It is also called glycosylation. So glycosylation takes place in Golgi apparatus. Addition of carbohydrate to the protein, or it might be the lipid, that we call it as glycosylation. So these are the two major functions of the Golgi apparatus. So if you take some questions related to Golgi apparatus, which of the following is not the function of Golgi apparatus? Not the function. It is involved in processing and shortening of glycoprotein. It is also involved in lipid metabolism, also involved in carbohydrate metabolism, but it is not involved in protein metabolism. Amino acid metabolism. That is not the functioning of Golgi apparatus. Amino acid metabolism does not depend on the cell organelle. Then coming to the second one, name the complex polysaccharide which does not synthesize in the Golgi apparatus. It is the starch that is not synthesized inside the Golgi apparatus. It will be stored in the cytoplasm after its synthesis. Hemicellulose pectins, glucose aminoglycans, all these are synthesized, or the modification takes place in Golgi apparatus. Then taking the third question, this is the function of glycosylation. Glycosylation that takes place inside the Golgi apparatus just to increase the stability of the protein. So glycosylation means addition of carbohydrate to the protein. It increases stability, so the answer should be B. The transfer vesicle from rough endoplasmic reticulum fuses with the region of Golgi complex at which phase. So for example, this will be the rough endoplasmic reticulum. This is the vesicle that is released. Then this represents the Golgi apparatus, the convex region. This vesicle fuses with this the convex part. That convex part, what we call it as cis phase. We call it as cis phase, so that answer should be C. Then the region where the vesicle has been released, that we call it as the trans phase. We call it as trans phase. Trans phase is also called maturation phase. Then cis phase is also called a forming phase. So the right answer will be C. So these are all some questions related to the the Golgi apparatus as well as endoplasmic reticulum. Regarding lysosomes, MCQs as well as the vacuole, that I'll discuss in the next video. So if you have understood these concepts, then like this video. I'll discuss lysosome as well as the vacuole in the another video. Thank you.